Okay, well, thank you for that kind introduction, Neil, and good morning, ladies and gentlemen. I feel honoured to be invited to participate in this very special meeting. I'm a huge fan of UK NEQAS. I think it has an enormously important role in education and support, as well as being a world-leading proficiency testing organisation. So it really is a pleasure for me to be able to participate. Neil's been through all that. I mean, really all I wanted to point out is that I've done a bit with UK NEQAS, admittedly in clinical chemistry. I've got a feel for international laboratory medicine, and as he said, I'm really here today on behalf of JCTLM, more of which will follow. I think I'm also here to be thought-provoking. This is an area I suspect that will be new to almost everybody sitting in the audience. So this is the outline. Let's start with a little bit of background. Strange slide. If I was to ask everybody sitting in this audience to tell me the time using your commonly used event, whether it's a watch or a phone or whatever, you'd give me an answer that was remarkably similar with a very, very small measurement uncertainty. If I was to take out of my pocket a pencil and I was to pass it to everybody here and I was to ask you to measure its length and to weigh it using commonly available tools, you would get very, very similar answers with a very small measurement uncertainty. And if I was to go to every one of the 32 countries currently in the World Cup and to ask them to do the same, they would similarly get very similar results with a very small measurement uncertainty. Now, how is that? That's not by chance, is it? That's clearly because we have traceable measurements in everyday things. So weight, length, time, temperature would be examples where there is global agreement on standardization. And the whole world can plug in very easily to that system. Just give you one example of that, and that's in the area of mass. There is a thing called the kilogram. It lives in the International Body for Weights and Measures, the BIPM, which is just outside Paris. And that is the international prototype for standardizing the kilogram. Individual countries then produce their own versions of it that are benchmarked against that international standard. And then through a series of calibration laboratories and mass standards and so on, we get to the balances that you might use in your kitchen, all of which are traceable back to that kilogram that's sitting in Paris. And this is really where the area of metrology comes in. Metrology is the science of measurement. This is the institute just outside Paris, the International Bureau of Weights and Measures, and there is a picture of the kilogram. And I have seen the kilogram. I feel like I ought to put that on my CV. And if you go to local museums and so on, this is one close to me, where I live. And here we have local weights and measures, and there you can see for length, for mass, and for volume, we have the local weights and measures, all benchmarked against these international standards through traceability. And they're underpinned by Système Internationale, or SI units, and here you can see the same parameters with their SI units, and these SI units underpin our scientific, manufacturing, and technological civilization. All fairly obvious, isn't it, when you think about it? So what about laboratory medicine? Well, we know it's difficult, don't we? No matter how you divide up laboratory medicine, it's difficult to get two methods to give you exactly the same result. And we know why that is, but the patients don't. And I've been to meetings of the public or the patients, and they are astonished to think that two different methods trying to measure the same thing on the same sample can actually get different results. And their view is very simple. They just say, fix it. Now, obviously organisations like UK NEQAS is doing its bit and helping enormously to harmonise the results, to reduce between method variability. But there are some fundamental things here that come into play as well. And that's perhaps what I'm going to try and take you through in terms of traceability in laboratory medicine. Let me just give you one example of what potentially can go wrong. So Haemoglobin A2 is a normal variant of hemoglobin A that consists of 2 alpha and 2 delta chains. It exists in small amounts in all adult humans. Its biological importance is uncertain. HbA2 concentration may be increased in beta thalassemia or in people who are heterozygous to the beta thalassemia gene. There are many clinical practice guidelines around the world for thalassemia that link diagnosis to target HbA2 levels. For example, the UK NHS Sickle Cell and Thalassemia Screening Programme states a national recommended cutoff for HbA2 of 3.5% has been set as the action point for the diagnosis of carriers of beta thalassemia. Very clear, very specific number actually given there. So what's the current performance of HbA2 in EQA? And here's some figures from UK NEQAS with permission. And obviously I've chosen a sample. Here's your 3.5% cutoff. There's the all methods distribution. But you can see, depending on which particular individual method you're using, that sample from that patient could be either positive or negative for a diagnosis of beta thalassemia. So we are potentially giving misleading clinical information because of the variability that exists between our methods. And there are hundreds of examples across laboratory medicine where this kind of potential clinical misleading can be produced because of the variability between the results. So we can conclude, I think, that the between-method variability of HbA2 methods at the clinically important cutoff is such that misclassification will occur. And back in 2012, this paper in the International Journal of Lab Haematology stated a poor alignment of routine methods for HbA2 measurement was found. The need for better standardization of HbA2 measurement procedures was underlined. Suffice it to say now that IFCC, in collaboration with the International Council for Standardization in Haematology, is working on the problem to try and produce the between-method variability for HbA2. So the status of HbA2 at the moment is unsatisfactory, but a collaborative project is underway to improve the situation. Now, I suspect from your own fields, you could come up with examples that show exactly the same challenge that faces all of us. So how can we reduce between-method variability to get more comparable results? Well, clearly, EQA, efficiency testing, has an enormous role in terms of monitoring performance, trying to see how the different methods align against one another and working with the manufacturers maybe to try and help reduce between-method variability. But sometimes there is a fundamental problem, and that lies in the design of the methods themselves. No matter how much good EQA you do, sometimes you simply cannot get methods to agree because they have fundamental differences in design, and this is where calibration and traceability to a common reference system may come in, and it gives rise to the whole concept of standardization and harmonization in methods in laboratory medicine. So that takes us into the area of traceability in laboratory medicine. A couple of slides now that how you might explain traceability in laboratory medicine to a member of the general public. And I've used these slides with the general public and they kind of seem to work. Of course they're oversimplistic, but it just sets the scene, and then we'll do a little bit more scientific a few slides on. So what is traceability? It's how we get the right answer. What is the right answer? It's an accurate answer, or the answer that we would get with the best method. How do I know if my result is traceable? The method is being compared with a better method and adjusted to give the same result. So that last one up there is really kind of giving us a pointer towards where we're going with traceability in laboratory medicine. And of course, in a nice little cartoon style, what we're trying to move is from there to there. Now, EQA can help us to move from there to there, but if there are fundamental design differences between methods, then perhaps we need a different approach to try and get a harmonization of results. results. And measurement traceability helps to make results the same anywhere, anytime. Remember back to the time and the temperature and the mass and so on. That, in a sense, is the ultimate aim of what we're trying to achieve. And final slide that perhaps aimed at the general public. You can think in terms of standardization. Here you've got 6 results. If you have a standard, then you can align each of those results against the standard, and with some careful work, perhaps particularly in the area of calibration, you can get all of these results to give you the same result. That's the concept of standardization. If you don't have a standard, then perhaps you're in the area of consensus, which gives you the concept of harmonization. But of course what you can do, and what I think increasingly is happening in laboratory medicine, is that you can get the world to agree on a conventional calibrator, assign a value to it, and then you can start moving these results together by aligning them to an arbitrary standard that you've given a value to that applies across the whole world. And that we'll see a little bit later into this talk. So that was the easy stuff as far as the general public are concerned. Let's get a bit more scientific now. And here we start running into rules and regulations. The IVD directive of 1998 stated, amongst lots of other things, that the traceability of values assigned to calibrators and or control materials must be assured through available reference measurement procedures and or available reference materials of a higher order. So that's a requirement in the IVD directive. And you say, well, a directive is advisory. But of course, we've moved on from then, and now we have the new IVD device regulation, legally binding regulation, published on the 5th of May last year and entered into force just a few weeks later. And this IVDR will become applicable 5 years after its entry. So in 2022, this will be a legally binding requirement, mainly, I have to say, on the IVD manufacturers. And the IVDR supersedes the IVDD, including changed classification rules and requirements for conformity assessment. It's intended to strengthen the current approval system for in vitro diagnostics. And the key paragraph in this one actually takes the IVDD a little bit further, because what it says is where the performance of devices depends on the use of calibrators and or control materials, the metrological traceability of values assigned to calibrators and or control materials shall be assured, shall means must, be assured through suitable reference measurement procedures and or suitable reference materials of a higher metrological order. Where available, metrological traceability of values assigned to calibrators and control materials shall be assured to certified reference materials or reference measurement procedures. That's pretty clear, and it's very demanding, and it's now less than 5 years away. So how are we going to be able to respond to that? Just before we get there, why is traceability a requirement? This comes out of the IVD directive. Obvious from what I've said so far, non-traceable IVD measurement systems may lead to a lack of product control, non-comparable measurement results, incorrect patient diagnosis and treatment, and uncertainty of compliance with the IVDD, now IVDR. So what is traceability in laboratory medicine in more scientific terms? I gave you a very easy one for the public, but in scientific terms, metrological traceability is the property of a measurement result, which can be related to a reference through a documented, unbroken chain of calibrations, each contributing to the measurement uncertainty. That's pretty stiff language, isn't it? But what we've got there is we've got results that can be related to a reference through a chain of calibrations, each contributing to the measurement uncertainty, or the degree of the imprecision, if you like, of measurement that goes with each level. And I'll show you in the next slide how this all ties together. So traceability requires both reference materials, ideally certified at global level, and reference measurement procedures, easier word is methods, in which they're actually used. So you need both materials and methods. Now for structurally simple measurands, it's another horrible word. Let's use analytes. It's not strictly the same, but you know what I'm talking about if I talk about an analyte. It's possible to get pure substance primary reference materials. So let's take glucose. You can get pure glucose. Okay, really easy. But of course, for more complex measure ends, things that you're dealing with, pure substance may not exist, let alone be available. Now, primary reference measurement procedures are usually based on physical methods. So we'll show in the next slide how this might fit together. For example, isotope dilution mass spectrometry is often used to assign a specific, absolutely accurate result to a reference material. So we have a hierarchy of reference materials, which we can think of, if you like, in terms of calibrators, and reference measurement procedures, which we can think of in terms of methods. So what we have is a routine laboratory procedure which has been produced by a manufacturer through a series of other measurement procedures, and ideally back to some primary reference measurement procedure that might be based on physical technologies such as mass spectrometry. And then in terms of reference materials, you have the product calibrator, which is in the method used by the routine laboratory, and then a series of other calibrators, ideally going back to a primary reference material, pure substance if it exists, and a primary calibrator, which might be that pure substance in, say, plasma. And that ideally should be SI traceable. And this is a hierarchy going from low to high in terms of metrological traceability. I hope that makes sense, and I hope that this slide helps to make it more sense. So here you've got the same results. You've got the patient result obtained with a routine laboratory method that has a product calibrator. And so on, you go back through a series of methods and calibrations, starting in the routine lab, then in the IVD method manufacturers, and ultimately, if it's possible to do it, through a metrology institute or a reference laboratory that might produce primary reference measurement procedure. And if you can have that unbroken chain from there to there, then you have full metrological traceability. And you can see increasing. At the same time, your measurement uncertainty or imprecision of measurement moves in the opposite direction. And that makes sense. So you've got the ultimate method here, using the pure material, and as you come down this chain, the measurement uncertainty gets greater. Hope that makes sense. That's the theory of metrological traceability. And you can put it in a slightly different way, and this perhaps is where it starts to become a little bit more relevant to you in LI. So here you've got your ideal situation of a primary reference material, a primary reference measurement procedure, primary calibrator, secondary reference material, and eventually you end up a secondary calibrator that then goes into the manufacturer's selected method. If you haven't got your primary reference material, you might produce an international conventional calibrator to get your calibration material. Maybe you can't do that, in which case you may just have an international conventional method. You say, okay, well, let's choose that method as the benchmark. And we'll benchmark everything against that method. We don't know whether it's the perfect method, but we'll all agree that's the one that we'll focus on. And you get to the calibration. Or you may, in fact, just start with an international conventional calibrator. That was, if you remember my slide of 6 points, saying let's put something in the middle there and benchmark all up against it, if you like, trying to harmonize methods. So you can do all of those approaches, and here you're going from higher order to lower order down the metrological traceability chain. And clearly the ideal is to try to get high up there as is possible to do, recognizing for many of the things that we do in laboratory medicine, that simply isn't possible. But better to get to here than not to do anything. That's really, I suppose, the message that I'm trying to get across to you today. So who are the stakeholders involved in achieving traceability? Well, you have internationally recognized expert clinical or laboratory committees, and I'd include UK NEQAS LI as an example of that, if you like, who define clinical decision values and analytical requirements. And then you have national metrology institutes, the British Standards Institute, for instance, would be one in this country, or professional bodies and societies who provide reference materials and higher order reference methods. So they produce the materials and the higher order reference methods. Then you have a global database of reference materials and methods, and these list what's available in terms of materials and methods, and I'll give you an example of that in a few minutes' time. And then you have standards institutes, and accreditation bodies that raise analytical and clinical targets to try and move us towards continuous quality improvement. And laboratory accreditation, which we'll hear about in a couple of talks' time, is an example of that. Then, of course, you've got the IVD method manufacturer who's being squeezed here, particularly by the IVD regulation that's coming in. And they produce the methods as traceable to a reference system when available. Then you've got the EQA provider, so if you like, this is where UK NEQAS LI comes in again, this time in its proficiency testing role, using commutable materials to monitor method performance. And finally, you've got the routine lab that selects the methods based on quality performance. So all of these people are involved, the stakeholders, in achieving traceability. And they've got to do it on a global level. So it's hugely challenging if we're to actually be able to pull this rabbit out of the hat. I mentioned the word commute, let's just go back to it. The word commutable here, use commutable materials, and this is aimed at the EQA provider, amongst others. What does commutability mean? Well, in simple terms, commutability, if you take a set of clinical samples and you measure, get a result by two different measurement procedures, there will be hopefully a straight line relationship between them. It may not be on a one-to-one gradient, particularly if the methods are giving vastly different results, but it should be a fixed relationship between those two methods. And if you take reference materials such as EQA materials and you measure them with the same two methods, then hopefully they will sit on the same line. If they sit on the same line, then those reference materials or EQA samples are deemed to be commutable. There's a lot of science gone in the last year or so to say how far off the line should they be before they're deemed to be non-commutable, but let's not go down that route just at the moment. It's just to give you the concept of what commutability actually is. Okay, so I've introduced to you the concept of traceability in laboratory medicine. I'm here on behalf of JCTLM, and you've probably never heard of it, so I should tell you a little bit about JCTLM. It's formed in 2002 as a direct response to the IVD directive. Try and get the world working together to manage the requirements of the IVDR directive. It had three organizations at its start. The BIPM, so that's that international body for weights and measures. Intergovernmental Treaty Organization for Measurement Standards, holds the kilogram. Okay. Then IFCC was the International Non-Governmental Organization for Professionals in Laboratory Medicine, and that's how I get involved through the Global Body for Clinical Chemistry in Laboratory Medicine, one of the leading bodies for laboratory medicine at international level. And then ILAC, which is the International NGO for Accreditation Bodies, so CPA as was, CAS as now is, actually belongs to ILAC as one of its members. Now, since then, JCTLM has grown, 47 members, 18 countries, and they comprise national metrology institutes, such as the BSI, EQA providers, several branches of UK NEQAS are involved, professional bodies, and IVD manufacturers. And we have to grow that 47 considerably before we can claim we have complete global reach. What does JCTLM do? Well, fundamentally, it maintains a reference measurement system database, which you can access, and I'll show you some slides on that at the moment. And it coordinates a nomination and review process for getting materials or methods entered onto that database. It contributes to ISO technical standards, particularly on reference systems, and it's currently revising two of these standards. This one here is on metrological traceability, the fundamental standard, and this one here is on international reference laboratories. And it's introducing new documents on commutability and on method harmonization, both of which I've touched on slightly in the last 20 minutes or so. Holds a biennial scientific meeting. But in addition, a bit like UK NEQAS, it provides educational materials to support an understanding of the importance of traceability in laboratory medicine. So it has an educational support role as well as holding the database. This is its structure. I don't really want to go into it in great detail, but You've got national and regional members, stakeholder members, all fitting into an executive committee and a secretariat. And then this bit here deals with the database, looking at all the materials and methods that go on it. And this bit here really deals with the education and the support for promoting the role of traceability. You look at the database, you go to the home page, you can say, well, I want to look at higher order reference materials or reference measurement methods or procedures, or maybe services. These are the international reference laboratories. You dot on that one, you put in what it is you're looking for, you click, and you can search the database very easily. This is completely free. Anybody is actually able to do it. And if you do, and you do something simple like looking at glucose, so this is glucose in frozen human serum. Here's an available reference material from Singapore, and a lot of information about it in terms of the assigned value, the level of uncertainty, which is really usually very small, something on commutability, and then what units it's in. And if you look at a method, this is the NIST method from the United States for serum glucose. It gives you, again, a whole pile of information about how it was done. This is an isotope dilution gas chromatography mass spectrometric method, primary reference measurement procedure, gives you the applicable range, expected uncertainty, has a publication to support it, and it's listed. Now, all of these entries are benchmarked against these 3 international standards. So you've got one for measurement procedures, that's the methods, you've got one for reference materials, and you've got one for reference measurement laboratories. So when applying to have something put on the database, the producer of the material or the method has to say how they comply with these 3 international standards. So what's the current status? Well, JCTLM, as of today, contains 302 certified reference materials, 188 reference methods, and 161 reference measurement services. That sounds impressive, but the estimate is that there might be 4,000 different methods in use in laboratory medicine, widest definition of laboratory medicine. So we're a long, long way from being anywhere near covering the scope of laboratory medicine. There is another database of reference materials, particularly held by the World Health Organization. That's the WHO Expert Committee on Biological Standardization catalogue of international reference preparations. And this has about 300 entries. There's very little overlap between the two. And I have to say here, there isn't much focus on traceability. This is much more a focus on let's select a reference material and let's agree that that's the one we're going to use, and let's assign a value to it, and then let's get everybody working together based on that assignment. And a lot of these materials are actually produced by NIBSC here in the UK. So even if you put the two together, we've got maybe 600 out of 4,000. So we've got a long, long way to go. And then just in terms of educational support, there's a special website that has a growing list of resources, publications, webinars, scientific presentations, scientific meetings. These are all available for use. There's some news updates and forms to apply for membership, and obviously a button to get directly to the JCTLM database if you want. So that's just a little bit about JCTLM. Now I just want to give you 2 case studies to show you what's actually possible. And I recognize that these are in relatively simple areas of laboratory medicine. The first one, cholesterol. Heavens, you can't get more simple than cholesterol. There it is, molecular weight 386. I did my PhD on cholesterol about 1,000 years ago. It's measured in all clinical chemistry labs, both as total and as high-density lipoprotein cholesterol. We know it's associated with increased cardiovascular risk. There's been an international standardization program since 1989, and it was one of the first analytes to have a reference laboratory network introduced in 2000. So it's been around a long time as an internationally standardized measurand or analyte. And of course there are clinical practice guidelines, lots of them around for existing coronary heart disease that link management to target cholesterol levels. Just one example here again in the UK, and it links the dose of statin that you're taking to the measurement of specific number for cholesterol or LDL cholesterol. So if it says 4, you've got to be sure that the methods can distinguish 4 from, say, 4.2 or 3.8 or whatever. So it's really important that you can actually hit a target if your whole patient management is being based on that specific number. Well, how are we doing? Here's some UK NEQAS data again, and these are patient donations dispatched on the day of collection, no preservative, and they were secondary reference methods. Values were obtained using the International Reference Measurement Procedure. And these are lots of different samples, and these are the between laboratory means from about 300 laboratories. And these are all individual samples, and you can see that across the biologically important range, the CV for cholesterol across all methods is 3-4%. So it's really pretty good, and that's as a result of this effort over many years to introduce standardization and reference methods. And so you can say that as a result of this, between method variability of cholesterol methods is at an acceptably low level. And what are the consequences of that? Well, the age-adjusted death rates from heart disease in the US fell by more than 50% over 25 years, and nearly a third of that could be attributed to improved secondary prevention using statins to lower serum cholesterol. So titrating statin against the cholesterol has had a dramatic result on the number of deaths from cardiovascular disease. And you can go even further than that as far as cholesterol is concerned. You can say cholesterol standardization has been shown to be cost-effective. It's relatively expensive to run a global standardization program, but you can see compared to the benefits to health that have been estimated, this is a trivial sum. So there's a huge potential saving as a result of standardization as well. So I think we can say, at least in the simple case of cholesterol, standardization improves clinical outcomes and saves shed loads of money for the healthcare system as well, which is the dream of every health minister in every country in the world. Second case study is a bit more complicated. This is hemoglobin A1C, so the hemoglobin molecule, and this has been established from major clinical trials as the key analyte for long-term monitoring of diabetes. And it's because you've got these green glycoprotein molecules attaching themselves to the hemoglobin. And method improvement followed an IFCC standardization that was introduced in 2004. Prior to that, the between method variability for hemoglobin A1C was about 20% and rising, 20-30% difference between methods. And as a result, it was very difficult to implement clinical practice guidelines. Now, the clever thing that they did in this method, this is a big molecule, as you can see. So what they did was they identified a signature hexapeptide that's unique to hemoglobin A1C, and they based the reference measurement procedure using mass spectrometry on that signature hexapeptide. Just looking at 6 amino acids linked together. And now there are reference laboratory network has been established that's around the world. And of course there are lots and lots of laboratory and point of care methods for HbA1c because diabetes is a huge business and there's lots of money to be made out of it, so everyone's got to have a method for HbA1c. Why is it important? Well, the DCCT trial, which is the Diabetes Control and Complications trial based here in the UK, actually showed that for every 1% that you can reduce the amount of HbA1c in the blood, you can reduce deaths related to diabetes by 21%, microvascular complications by 37%, and myocardial infarction by 14%. So huge clinical benefits from actually titrating your HbA1c, getting your diabetes under a better level of control. And so much so that an international consensus statement was issued in 2010 saying that results have to be standardized worldwide. There is a reference system saying that the IFCC method is the only valid anchor, and then lots of other recommendations about editors and how you report it and so on. And as a result of this, the IFCC has set up a committee on the implementation of HbA1c standardization. Gary John from Norfolk is the chair of that, and Gary's got a great job. He travels around the world trying to persuade international diabetes associations and international professional bodies that they should all unite behind this IFCC reference system and method that everybody should report HbA1c worldwide. It's an ongoing process, but Gary tells me in the last fortnight he's had a breakthrough in China. The whole of China, all however many billion people, will now be benchmarking themselves against this particular approach. approach to reporting hemoglobin A1C. What's the performance like? Well, it's not quite as good as cholesterol, but using the same approach, here you are around about 4 or 5% between laboratory agreement from, 349 laboratories. So it's pretty good, but not quite as good as cholesterol. But it's a lot better than 20 to 25% that it was before we started this exercise. And so as a diagnostic test, you can say it's a lot better, and it's a fairly reliable, as a monitoring test, you could say it's a lot better and it's a fairly good test. But that actually then persuaded the World Health Organization to say, well, okay, methods for HbA1c have now improved so much that in addition to monitoring diabetes, we can now use it for the diagnosis of diabetes. And it's now, in many countries, the first-line approach to the diagnosis of diabetes as well as to monitoring treatment. And this is this guideline that says HbA1c can be used as a diagnostic test provided that stringent quality assurance tests are in place and assays are standardized to criteria aligned to international values. The World Health Organization is recognizing the importance of international standardization and saying, okay, great, now we can use it as a diagnostic as well as a monitoring test. And you can then take it a little bit further because you can introduce models to set and evaluate quality targets for HbA1c using either biological variation or sigma metrics. And without getting too complicated about this, here is the 2 sigma, 4 sigma, 6 sigma line. These are all individual methods. Anything that lies under here is performing extremely well. These methods here are not performing so well, and it may not surprise you to know that many of these methods are point-of-care testing methods for hemoglobin A1c. So the pressure is on to get all of these methods moving into even better performance characteristics for the use in diagnosis and monitoring diabetes. Okay, coming towards the end, what's all this got to do with UK NEQAS LI? You can think, well, clinical chemists have done the usual trick. They've picked all the low-hanging fruit. All the easy stuff like glucose and cholesterol and creatinine and HbA1c and enzymes and simple proteins. And that's true. But if we think in terms of the long term and we think in terms of 4,000 different things that we're measuring in laboratory medicine, then these include things like nucleic acids, increasingly antibodies, cells, of course, and microorganisms. And these are much more challenging than these simple chemical entities. And if we express that in a slightly different way, we can say the challenge in terms of method standardization and harmonization is set out here. So we've got a number of methods, or if you like, is 0 up to 4,000, and we've got complexity in actually bringing about standardization or harmonization. And the small molecules, the simple peptides, where we were dealing predominantly with chemical variability, are the ones that we've gradually been ticking off. The challenge lies in some of the more difficult things like nucleic acids, antibodies, cells, and microorganisms. And here we're dealing with biological variability as well as chemical variability. So much, much more challenging. But challenging doesn't mean impossible. That's really the key to this. Now, as Neil said, I was invited, and I'm delighted to be invited to meet the steering committee for UK NEQAS LI last year, and we agreed potentially 3 things that UK NEQAS LI could do. First of all, it could become a JCTLM stakeholder member. It did that very quickly, no money involved, and that's just simply a means of communication, nothing more than that. We said it might be possible to have a presentation on traceability at your Congress. Here I am. And the third thing was to select an index method from within your repertoire that is of clinical importance with recommended action limits. So remember the HbA2 story, something a bit like that. Shows unacceptable between method variability in your scheme, and maybe consider it as nomination for an international harmonization project using this organization, www.harmonization.net, which acts as an international body to try and select projects for international harmonization efforts in the area of method harmonization more standardization. Since then, I've been doing a little digging around and attended a meeting fairly recently where we had people from, I'm not going to attempt to actually say the German, but PTB in Braunschweig in Bern. This is a National Metrology Institute in Germany, and they've been working for a number of years now on reference measurement procedures for the accurate determination of cell concentration, present status and future developments. This is from way back in 2012, so they've been working on this and continue to work on it. And they have now developed primary reference measurement procedures based on flow cytometric cell counting by impedance measurement. So this is where your physical methods coming in, impedance measurements. They've established them for erythrocytes, leukocytes, and thrombocytes, and the method is being validated at the moment for CD4 plus lymphocytes. And these methods are all described in DIN standards. Now, DIN is a German industry body, and so they're all described in German. So they're not necessarily known to the wider world, that these primary reference measurement procedures for cell counting, cell concentration, they do actually exist. In reality, I think it's fair to say none of the manufacturers are using them. Maybe none of the manufacturers know that they actually exist. Because we've got the disconnect here between the National Metrology Institute and, dare I say, the real world. Also, there's a body, again, you'll never have heard of this, but lots and lots of money, EU money, is going into an organisation called Euromet. MET stands for metrology, which again, science of measurement, if you remember. And there's a big project called BioCitrace, which has just published its summary for one particular traceability project, and this describes traceable nucleic acid, protein, and cell measurement procedures based on enumeration technologies. So there are now, again, through Euromet, BioCitrace, actually methods being developed and references provided. But once again, not a lot of people know that, because we've got the pure scientists that are involved in the metrology side and this chasm between the users of the methods and the patient community. So perhaps, if I could be impertinent enough to suggest to UK NEQAS LI that there might be an additional possible role for you as we look to the medium to long term, and that's to see if any of these new reference measurement procedures that are being developed could actually be something that you could think about and again develop a case study based on these to see whether it's possible to harmonize some of the methods that you're using, which are so vitally important to individual patients and their long-term health. Thank you very much for your attention. I hope that's given you something to think about.